Welcome, my dear students. Now we are going to have a quick revision on this chapter, microbes in human welfare. Okay, so coming to this introductory part, so we have these microbes everywhere. Everywhere we have the microbes inside the body, outside the body, in soil, water, everywhere. There is no place literally without any microbes. They can be used in this seen in thermal vents where temperature is as high as 100 degrees Celsius. And what are the different types of microbes? There are different types like protozoa, bacteria, fungi, microscopy, plant viruses, viroids, and prions, protein, infectious particles, etc. And so how we can study microbes? We can study microbes using culturing colonies. Fungi can be grown, bacteria and fungi can be grown in nutrient media, that is by culturing. Such cultures are useful in the studies of microbes. And these are the different types of microbes. So we have this bacteriophage. This is bacteriophage, and this is a virus. That is the virus. This is a rod-shaped TMV virus. This is a bacteria, rod-shaped bacteria. This is spherical shape. This is again this rod-shaped bacteria, spherical bacteria, rod-shaped bacteria, etc. And this is adenovirus. This adenovirus, this is adenovirus, which causes respiratory infections, upper body infections in children. And this is, as you know, this is a bacteriophage. This is our common, this TMV. And these are the culture plates where these microbes are grown. These are the colonies of microbes, etc. Now, as you know, these microbes cause many diseases in plants and animals, but we should not think that they are harmful to human beings. But no, they are actually in different ways, more than harmful effects. They are having beneficial effects. Many of the microbes having beneficial effects. And there are nearly 9 or 10 points like this. One is microbes in household products. Second is microbes in industrial products. Third is this microbes. These are all industrial products. Microbes in sewage treatment. Then microbes in biogas production. And lastly, we have microbes as biocontrol agents. And these are the main headings. And these with the subheadings, there are several subheadings also. And the subheading, including the subheadings, it will come to about 9 or 10 points that we have to make a quick revision. Okay. Now these microbes as household products. As household products, okay. As household products, microbes have been used from time immemorial. A common example is this lactic acid bacteria. Lactic acid bacteria, which is lactic acid, which can milk into curd. And lab produces, how this lactic acid bacteria converts, they are actually coagulating the milk protein, and the milk protein is converted into curd. And when this milk protein is converted into curd, it is increasing the quality of vitamin B12, and this can be used to check bacterial diseases since it causes an acidic region. Then this another is this duff for making idli and dosa. They are actually fermented by bacteria, and the carbon dioxide gas produced as a result. So there is a puff appearance. And another similarly, this for making bread, making baker's yeast, Saccharomyces cerevisiae, etc. And then fermenting toddy, and this one of the ancient food material like cheese. And the quality of the cheese, the flavor and the taste of the cheese is made by the large holes in cheese due to the production of large amount of carbon dioxide by bacteria, Propionobacterium shermani. Okay. That is household products. Next is industrial products. In industrial products, there are mainly two examples. One is this beverages, and second is this antibiotic. Both these beverages and antibiotics are used in steel, huge fermenters like huge tanks. They are known as fermenters. And in huge tanks, these are the fermenters. where this baker's yeast, here the same yeast, baker's yeast is used for the yeast which is used for making bread is brewer's yeast, where it is fermented. This fruit juice, malted fruit juice or malted cereals or fruit juices are fermented and it is converted into alcohol. And there are different two types of mainly alcohol. One is used without distillation and with the distillation, etc. And this antibiotic, as you know, the first antibiotic that is penicillin. The antibiotic was discovered by this Alexander Fleming, etc. And actually they are meaning against life, but with regard to human being, they are pro-life. And antibiotic, the penicillin, and that was discovered by Alexander Fleming. And when he was culturing the Staphylococcus bacteria, he found in some of the places of this culture medium, Staphylococcus was not growing. And he found that there is a fungus. That fungus was Penicillium notatum, and it produces some chemicals, so it inhibited the growth of Staphylococcus. And after Second World War, this was extensively used for the wounded soldiers, and Fleming, Chain, and Florey. They were actually Chain and Florey popularized the penicillin, and both these three of them, Fleming, Chain, and Florey, they was awarded Nobel Prize after the Second World War. Now, this next is bioactive chemicals, enzymes, and other bioactive molecules produced from these microbes. There are many organic acids, alcohol, enzymes, etc. can be produced, for example, on the industrial basis, not only in a household basis, it is industrial basis. For example, Aspergillus nigra fungus, and we have citric acid, Acetobacter bacteria, acetic acid, Clostridium botulicum bacteria, which produce butyric acid, Lactobacillus produces, a bacterium produces lactic acid. So this bacteria, fungus, etc., you must study by heart. Then this is Saccharomyces cerevisiae, commercial production of ethanol, that is another industrial use. And many enzymes can be, for example, lipases can be used in detergents to remove oils from our clothes, especially from people working in what is called oil-based industries, workshops, etc. Another is the fruit juices. Fruit juices, when we got the notice that bottled fruit juices from the market, they are clearer than those made at home. In home, we do not use any such enzymes, etc. So the fruit juice it is thick and very pulpy, whereas fruit juice we obtain, say mango juice we obtained from market, it is clear, just like water, because in industry they are using clarified using pectinases and proteases. So all the protein, pectin, etc. will be removed, and that gives a clear solution. Whereas in houses we don't use such pectinases and proteases, etc. And there's another medicine, streptokinase, produced by Streptococcus by modified genetic engineering, is used as a clot buster for removing clots from the blood vessels of patients having this block of heart. Another bioactive molecule, that is cyclosporin A, that is used as an immunosuppressive agent for organ transplantation patients, and it is produced from this Trichoderma falciparum, very common fungus seen in the soil. Another is statin, produced by Monoscus purpureus, which can be used to lower blood cholesterol lowering agent, and it is competitive inhibition, as you studied in biomolecule, to reduce the production of cholesterol. So that is with regard to these microbes in industrial production. Next is microbes in sewage treatment. Sewage means all the wastewater, a large quantity of wastewater are generated every day in cities, towns. A major component of this wastewater is human, etc. And it contains many microbes, most of which are pathogenic. So it is not safe to pump or safe to just open the sewage into drinking water, rivers or ponds, etc. So we have to treat it. First of all, sewage has to be treated using sewage treatment plants and make it less polluting, not 100% pure, to make it less polluting. And in sewage treatment plant, we have different methods, like this is the sewage treatment plant. And in sewage treatment plant, we have two methods. The first method is known as primary treatment. Second method is known as secondary treatment. or biological treatment. It is biological. Here microorganisms are being used. In primary treatment, there are again two processes. One is filtration and second is the sedimentation. And by filtration, all the floating materials, sedimentation, all the floating materials are removed by filtration. By keeping some comb-like structures, all floating things are removed. And this water is slowly flowed into a huge tank where water is slowly flowed so that and the time is allowed for sediment. So all the solid materials like grit means small soil particles, then pebbles, etc. And all such solid materials will be deposited in the bottom after sedimentation. This is known as primary sludge. And since this contains many soil and solid particles, this can be used for this land filling, etc. And the supernatant liquid from this after sedimentation is pumped into a tank known as aeration tank. So after this, this is the primary treatment is finished. Now here onwards it is secondary treatment. First tank is aeration tank where air is pumped into. So there is plenty of aerobic bacteria of the sewage with good fungi and aerobic bacteria. They grow immensely. They just grow. So that huge amount of this aerobic bacteria and fungi are formed, and sometimes they form some flocks like they form some solid, semi-solid these fungi. The bacteria, they colonize between the filaments of fungi, so they form some semi-solid or solid-like material known as flocks. And as this fungi, aerobic bacteria and fungi grow, their food material, their energy deriving material is actually this organic waste, organic pollutants, organic waste in the sewage water. So this organic waste material is slowly depleted. So we have to see this. Now and then we are testing BOD. BOD means biological oxygen demand, by which this when biological oxygen demand is low, that means organic pollution is minimum. When there is a direct proportion, when BOD is low, organic matter pollution is low. When BOD is high, organic matter pollution is high. So here and now and then we usually take this test of this BOD, and BOD is very low. That means organic matter is almost completely exhausted, or this organic matter pollution is very low. Now we pass this water into another tank known as settling tank, where these flocks are settled, and a few quantity of flocks is taken as an inoculum into the first chamber, aeration chamber, so that the organic matter depletion will take place. The reduction of this, what is called this decreasing the quantity of BOD will take place. BOD value will become low very quickly. So the settling tank flocks are settled. From this, it is transferred into anaerobic sludge digester. Where anaerobic bacteria eat away all these aerobic bacteria and fungi, so that some gases like hydrogen sulfide, methane, and carbon dioxide are produced, which can be used as this what is called as biogas. Now after this, water is almost less polluted. All the organic matter is being removed, so this is less polluted. Now only less polluted water. Main quantity of pollution is over, so less polluted water is pumped into this water channels. So this is sewage treatment plant. Next is this Ganga action plant and Yamuna action plant. This is taken up by this government of India, this Ministry of Environment and Forest, Ganga action plant and Yamuna action plant, to clear up our sacred rivers. Then this microbes as a production, biogas production. For the biogas production, the mixture of biogas mainly containing methane produced by the microbial activity, which may be used as a fuel. Then the microbes produce different types of gases, and by the end product of growth and metabolism. And the gas produced depends upon the substrate which is used by the microbe. So the gas produced depends upon the type of microbes and the organic substrate that is being utilized. For example, for fermentation of duff, cheese, and these beverages, alcoholic beverages, the gas produced is carbon dioxide. But at the same time, there are certain bacteria which are growing anaerobically on cellulosic material, which produce large amounts of methane and carbon dioxide and hydrogen. And such bacteria are known as methanogens. Methanogens means bacteria which produce methane. And such a methanobacterium is a typical example. And this is usually present in the rumen of cattle. And it is also present in the dung. And using this dung contains plenty of this methanobacterium. And using that dung, we can produce methanobacterium in the dung, we can produce biogas. And the biogas plant, this is a biogas plant. This plant is devised by Indian Agriculture Research Institute, IARI, and Khadi and Village Industries Commission, KVPY. This is a huge tank, 10 or 15 feet deep, and we add this vegetable matter and all animal matter, etc. here, along with some dung and water, and it will go down into this digester, where this dung, the bacteria, methanogens containing in the bacteria dung, digest all this vegetable and cellulosic material, etc. And methane and these gases, methane, carbon dioxide, and hydrogen is collected. And slowly, this is a lid. Lid is movable. This lid will go up. And from the lid, we have this outlet pipe is connected, which can be used, directly connected to a burner. And so as more and more this digestion takes place, some slurry, some waste material will be collected. That waste material is collected in this sludge, and which can be used as a fertilizer. So that a cheap source of this biogas is produced, which can be used for burning fuel as a fuel, or as what is called used for this lighting purposes, etc. And these biocontrol agents. Biocontrol agents means biological methods for controlling plant diseases and pests. We are now using huge quantity of insecticide, pesticides, etc. And these are extremely harmful to human beings. It causes pollution, etc. So by using these biocontrol agents, biological agents to control pests, etc. We can reduce the use of insecticides, pesticides, etc. And two such examples are this, ladybird, which will remove all these aphids, and dragonflies, which can remove mosquitoes, is a good example. Another example is microbial control agent that can be induced in order to control butterfly caterpillars is the Bacillus thuringiensis, written as BT. And this is available, now it is available in small plastic packets, which can be mixed with water, and when it can be sprayed on the leaves. And when these larvae eat these leaves, these bacteria also goes into their intestine, and these bacteria will later on, these genes, BT genes, will later on, what is called, they will break, or they will make holes in their intestine, and the caterpillars will die. And the bacterial disease will kill the caterpillars, but leave other insects unharmed. And now these BT genes are transferred genetically, what is called genetic engineering into cotton, and such a cotton is known as BT cotton. If this larvae eat this BT cotton leaf, then naturally the intestine will be broken, and insects, these larvae will be dying. Another example is biocontrol agents is this trichoderma. Trichoderma used in the treatment of plant diseases is the fungus trichoderma. It's a free-living, trichoderma species is free-living fungi that are very common in the root ecosystems. They are affecting biocontrol agents for several plant pathogens. Next, another this biocontrol agent is baculoviruses, which are pathogens which can kill the attack of insects and other arthropods. And these baculoviruses used biological controllers are in the group of nucleopolyhedroviruses. Nucleopolyhedroviruses, baculoviruses, which can be used to attack or kill insects and other arthropods. And these viruses, very important point, are excellent candidates for species specific. That will destroy or that will kill only that particular species. So species specific means narrow spectrum. Whereas insecticide, just like insecticide, they have narrow, whereas they have some killing some insects, but only narrow spectrum means that particular insects only. Whereas the chemical insecticide kill all types of organisms. So they are broad spectrum. So these viruses, nucleopolyhedra group of viruses, they are narrow spectrum, species specific insecticide. They have shown to no negative impacts on other plants, mammals, or birds or fishes, or of non-targeted insects, pests, etc. They have no impact, whereas they will kill only that particular insects, etc. This can be used when a particular region is highly ecologically sensitive. We cannot use any chemicals there. So they are such an ecologically sensitive area, other organisms will die. Whether if there is some infection of some arthropods or insects, we can introduce this polyhedro group of viruses, baculoviruses, which will kill only that particular insect and or arthropods, etc. So that is used in integrated pest management programs. Next is microbes as biofertilizers. Microbes as biofertilizers. This present day lifestyle environmental pollution is a major concern, and these chemical fertilizers are increasing, and it causes this significantly to this pollution. Nowadays we are using organic farming. We are switching over to organic farming. What is meant by organic farming? It is biofertilizers. And what are biofertilizers? That are organisms. It is not chemicals. It is organisms that enrich nutrient quality of the soil. The main source of biofertilizers are bacteria, fungi, and cyanobacteria. They are used especially in the root nodules of leguminous plants. And rhizobium, we have already studied symbiotic nitrogen fixing. Rhizobium we have studied. In a similar way, other bacteria can fix atmospheric nitrogen, like Azospirillum, Acetobacter. They are free living and enriching the nitrogen content of the soil. Fungi are also known to this fungi or symbiotic association with the mycorrhiza. Mycorrhiza are this fungal association of, it is symbiotic association of fungus and roots of higher plants. This is the symbiotic association that is known as mycorrhiza. And common fungus is glomus, which form mycorrhiza. The fungus do this fungal of mycorrhizal symbiont, they do 2 or 3 beneficial effects. Association absorb phosphorus from the soil and pass it to the plant. Phosphorus, that is the first use. Phosphorus is a mineral which is the plants obtain phosphorus in very little quantity, but this fungus of this mycorrhiza can absorb phosphorus and from the soil and pass it on to the plant. So another benefit is resistance to root-borne pathogens. That is second benefit of this mycorrhiza. Resistant to root-borne pathogens, tolerance to salinity, that is the third benefit, and drought, and an overall increase in plant growth. That is the fourth one, plant growth and development. Okay. Another thing is this cyanobacteria. Cyanobacteria are autotrophic. They are living in aquatic conditions. Anabina, nostoc, oscillaria in paddy fields, which can improve important biofertilizer. Blue-green algae add then this they can fix atmospheric nitrogen and add fertility to the soil and by which the plant production is increased. Another thing is when these cyanobacteria dead, the organic matter is deposited in the soil, that also increases fertility of soil. And biofertilizers are available, and one of such is azolla. Azolla contains this Anabina azole, which can be used, which can fix atmospheric nitrogen, and the plant growth can be enhanced very much. And so just as we make the quick revision, you just go through the important points. This is comparatively a simple chapter. So just go through the important points. I will just mention the important points only. Yes. Now, where does all these microbes are present? Microbes are present everywhere. They are present everywhere. That is the places where microbes occur, habit where microbes occur, and what are the different types of microbes. Now, next point is how microbes can be grown. They can be grown by culture, and you just go through these figures and study by heart the labeling, etc. And microbes which can be used as household products, lactic acid, bacteria, which can increase the B complex and as a checking diseases, and this duff for Italy and those, etc. And fermented baker's yeast, then toddy, and this one of the oldest food item, cheese, and this industrial product like beverages and antibiotics. And beverages, two types of beverages. Beverages is Saccharomyces cerevisiae is acting on bread making, so commonly brewers yeast, and they are fermenting malted cereals and fruit juices, and ethanol is being produced. And two types of these beverages, one is without distillation and with distillation, etc. Antibiotics against life, but with regard to man, it is pro-life. And first antibiotic penicillin, Alexander Fleming, Staphylococcus, etc. Then Chain and Florey popularized, and later on Fleming, Chain and Florey were awarded Nobel Prize in 1945, etc. Then these bioactive molecules, there are several microorganisms from which several industrial level products can be Aspergillus niger fungus, citric acid, Acetobacterium acetate, that is acetic acid, Clostridium botulinum bacterium, butyric acid, and lactobacillus bacterium lactic acid. Then this Saccharomyces, alcohol is, ethanol is produced, and lipases to remove this oily stain from laundry. Then this fruit juices, they are clarified using pectinases and proteases. Streptokinase from Streptococcus by genetic engineering, which is a clot buster. Then this immunosuppressant, cyclosporine from Trichoderma polysporum, and statins from Monoscus purpureus, etc. Then sewage treatment. What is sewage? Sewage contains wastewater, a lot of human excreta, a lot of microbes, which most of which are pathogenic, and it is the sewage water must be treated through sewage treatment plants and make the water less polluting. And who is actually doing this sewage treatment? We are only giving facilities for sewage water collection, etc. Actually, it is done by heterotrophic microbes naturally present in the sewage. Heterotrophic microbes naturally present in the sewage. And this primary treatment, secondary treatment, biological treatment, aeration tank, settlement tank, and anaerobic sludge digesters, etc. And in the anaerobic sludge digester, gases during digestion of aerobic bacteria produce mixture of gases like methane, hydrogen sulfide, and carbon dioxide, etc., and which can be used as a biogas. And now this sewage treatment is now this Ganga action plant and Yamuna action plant. And biogas. Biogas, the main points is the mixture of gases containing predominantly methane, and the gas produced depends upon, the type of gas produced depends upon the type of gas produced depends upon the microbes and the organic substance which is being used. Carbon dioxide is produced during beverages production, duffs, cheese making industries, etc. And some bacteria growing on cellulosic material produce methane, carbon dioxide, and water, and hydrogen. Such a bacteria are called methanogens. And a typical example is methanobacterium. And this methanobacterium is very commonly present in the rumen along with the dung of this cattle. And what is called this biogas plant. Biogas plant developed by Indian Agriculture Research Institute, IAR, and Village and Khadi Industries Limited, etc. So this next is microbes of biocontrol agents. So we can reduce the pollution of chemical pollution. We can avoid the use of insecticides, pesticides, etc. Then what are the different examples? Two examples. One is ladybird beetle, which can kill all these aphids, and dragonflies, which can remove these mosquitoes, etc. Which can be, another is the BT, which can be used to kill these butterfly caterpillars, bacteria, Bacillus thuringiensis genes, BT. And now this BT gene is introduced into cotton, so that is BT cotton, etc. Another biocontrol agent is this trichoderma, which can be free living, very common root, and they effective in controlling many plant pathogens. Next, another type of biocontrol agent is baculoviruses, which is coming under nucleopolyhedroviruses. These are narrow spectrum, species specific, narrow spectrum with the insecticidal, just like an insecticide. This can be used, but this the insecticide is broad spectrum. Several organisms will be killed, whereas this narrow spectrum means they will kill only that particular arthropod or insect. So this is used in an integrated pest management program. Then biofertilizers, slowly we are now using this to reduce pollution. We are switching over to organic farming, use of biofertilizers. And in biofertilizers, they are not actually chemicals, but they are organisms that enrich the quality of the soil. Biofertilizers are bacteria, fungi, and cyanobacteria, and root nodule formation in leguminous plant rhizobium, and free living azospirillum, acetobacter, etc., and which can fix atmospheric nitrogen. Another is mycorrhiza, association of fungus, symbiotic association of fungus with the roots of higher plants, and common fungus is glomus, and these beneficial effects of this mycorrhiza, the fungus can absorb phosphorus and it is given to the plant. It can show these benefits, other benefits like resistance to root-borne pathogens, tolerance to salinity, drought, and on overall growth, help in the overall growth of the plant. Then the cyanobacteria, which can free living cyanobacteria like Anabina, Nostoc, Oscillatoria, which can again fix up nitrogen and increase the fertility of the soil. Similarly, by the death and decay of these Anabina, Nostoc, and other blue-green algae, the fertility of the soil is also naturally increased. Then another common biofertilizer is azola, which contains Anabina azole, which can fix atmospheric nitrogen. And that way also, this azola is a pteridophyte. It also can be used as a biofertilizer. So this is comparatively a simple chapter, a comparatively a small and simple chapter. So you just remember all these points. Okay. So that is all with a quick revision regarding these microbes in human welfare. We'll stop here. So take care. Okay.